Today, we are going to be inoculating cultures to look at the ability of our organisms to digest polymers. So the first one that we're going to set up is a gelatin test. So this tube is going to contain gelatin, and we're looking for the ability of your microbe to break down gelatin. So gelatin is kind of like Jell-O. It's actually one of the components of Jell-O, where basically it's going to be a liquid when it is not chilled. When it's chilled, it will turn into a solid. So we're looking for, specifically for proteases, which are proteins that break down gelatin. They're called gelatinases. So for this experiment, we are going to inoculate our gelatin broth using a loop, just like we would do a normal inoculation. And basically, if your organism produces these proteases that break down gelatin, your gelatin tube is going to stay a liquid when we solidify it, or when we put this on ice. So that's your positive result. If your if your gelatin tube turns solid after putting it on ice, your organism is negative for these proteases. The second thing we're going to look at is the ability of your organism to break down starch. So in this test, we have a plate that contains starch. The starch plate has one blue and one green streak on the side. And we are specifically looking for the presence of the enzyme amylase. Amylase is the enzyme that breaks down starch. Some bacteria have this enzyme, some bacteria do not. So again, you sterilize your loop. You're going to pick up one isolated colony from your plate, and then to do our starch plate, we are going to make one streak straight down the center of your plate, about an inch or so long. So your plate will be streaked like this. We want to keep all of our culture concentrated to that one streak so we can look for any clearing in the starch surrounding the streak. If you were to streak for isolation, it would be very hard to see if your organism is producing amylase.